Is loud and clear? Yes, is my voice audible? Hello, Braj Kishore. Hello, Kanba. Hello, Sachi, Amala, Devanshi. Very good evening. Okay, thank you for the confirmation. So, I welcome you all to day 9 and last session of this rapid revision round series. Today, in this session, we will revise Unit 12 and 13. So, Unit 12 is about applied biology. And applied biology, although it's a mixture of many topics, but if you go through previous year question paper trends, you will find that the questions are mostly asked from agrobacterium mediated gene transfer process. So when you're preparing for Unit 12, make sure you revise this particular topic thoroughly. So are you all ready and excited to practice questions from Unit 12? If yes, then please write bring it on in your chat box, BIO in your chat box. Okay, that's great. I'm glad to see all the response coming. Thank you everyone for confirming. So I'll be giving you a few questions from Unit 12 first. We'll practice few questions from Unit 12 first, and then we'll move on to Unit 13. So let's get started. Here comes the first question on your screen. So question 1 is, given below is a schematic representation of the tDNA region of a transgene construct and the southern blot analysis using PST1 digested genomic DNA of 5 independent transgenic lines labeled as A to E, developed using the construct. The probe used for hybridization is shown as a black box below the construct, and UT is the untransformed plant. Now, based on the above data, which one of the following options gives the correct list of single insertion transgenic events? The options are on your screen. So tell me, according to you, which is the correct option? So let's see how quickly you solve this question. You have to keep in mind time also. The clock is ticking. So when you solve the question, also make sure that in how much time you are able to solve a particular question. Okay, Kaveri, very quick. Option 2, that's great. Amala, option 2, that's great. Let's see what others, according to others, what is the correct option, correct answer. Okay, Nandini, option 2. So far all are saying option 2. Devanshi, Manoj, option 2. Okay, that's great. I want you all to answer, okay? I want all the participants to answer. Don't hesitate, okay? Please try. Vishnu, Jaya, Murthy, Manoj, okay, option 2. So quick. Glad to see the answer. Very good. So let's see how is your preparation of Unit 12. This is the question that was asked in year 2020. Still, I can see all of you have not answered the question. Please try. I want everyone to try. I know you all can do that. Take your time, answer the question. To answer this question, you need to be familiar with Southern blot analysis technique, right? You need to be aware of transgene. What is transgene? Okay, so all have answered option 2. That's great. So let's check whether the answer is right or wrong. So basically, as I told you in the beginning, you need to be familiar with Southern blot technique, right, to answer this question. Now, Southern blot technique is the technique which was developed by E.M. Southern in year 1975. And using this technique, we can detect a particular DNA sequence in a DNA sample. So here on your screen is the Southern blot technique. And you can see that these are the main methods, the key steps of Southern blot technique. So first step is you need to have a DNA sample. You need to isolate the DNA and you need to cleave it with restriction enzymes. Now, when you perform the restriction digestion using restriction enzyme, you get a lot of fragments. Now, these fragments, DNA fragments, will be separated by performing electrophoresis. So you perform electrophoresis and separate these fragments. And then you perform blotting, where you transfer the bands from the gel to the nitrocellulose filter. Once the bands, the DNA fragments comes over the filter, you perform the hybridization. So key to this Southern blot technique is, Southern blot method is hybridization. We use a probe, radio label probe, and we add. So the probe, which is complementary to the DNA sequence, the DNA sequence of our interest, it will bind to it. And on performing autoradiography, you can see the DNA of our interest, right? Now, here in this question, they have mentioned that restriction digestion was performed using PST1 enzyme, right? Restriction enzyme. So if we perform the restriction digestion, you can see we'll get 3 fragments, right? So this is the first fragment, this is the second fragment, and this is the third fragment that we'll get. But the point that you have to focus here is, first, that we are performing Southern blot analysis, and second, we have to look for single insertion event. So obviously, when we perform restriction digestion with PST1, we'll get 3 fragments. However, on performing the Southern blotting, on performing the Southern blotting, on performing autoradiography, only those bands to which this probe has binded will be visible. So this is the catch here. Those fragments to which this probe has binded will only be visible. So on performing the Southern blot and on when you visualize these fragments, you will be able to visualize 2 bands out of 3, right? And as the question is about single insertion event, so now if we look at the bands, we'll find that one band is common, right? In the untransformed plant sample lane, we are getting one band, and this band is common for A to E lane also, right? But other than this, you can see some more bands. So one band is common, so that means we'll see one band for UT and 2 bands for the transgene if single insertion event has taken place. So that means you have to specifically look at the lanes in which we are getting overall 3 bands, 1 for UT and 2 for transgene. So if we look at the bands carefully, so basically you have to focus on number of bands here. And A lane, B lane, and E lane, there only you see 3 bands. And this indicates single insertion of the transgene. And this is the reason the answer is option 2. So I'm really glad to see all the answers. You all have answered correctly. Very good. Keep it up. Moving on to the next question. So here comes the next question on your screen. So this is the match the type following question. Please go through it, everyone. Make sure that you always prefer to answer or attempt these questions. Match the following type questions. So the question 2 is about match the following Vir proteins with their correct function during agrobacterium-mediated transfer of tDNA to plant cells. So you have to choose the correct option, correct combination. Now, why I'm asking you to attempt such questions, because usually they are asked in part C and they are very scoring. You can easily score 4 marks. If you know the function of any of the two Vir proteins, then also you can answer this question. So tell me, what is the answer here? Okay, Anshu, Nandini, Kaveri, Vishal, Kriti, Devanshi, all are saying option 2. That's great. I'm getting uniform answers today. Very good. Today in this session. It happens very less. But I'm really glad to see all your responses because this shows your preparation. Okay, Amala, Vijay, Sonali, option 2. Sonali 2. Okay. I want everyone to answer this question. Everyone should participate. How you will solve a question, such type of question, if it is asked in exam? Ankita, Pavi, option 2. Okay. Let's see if you have answered correctly or not. I'm giving you a few more minutes. Manoj, option 2. Okay. Advesha, option 2. Fine. Vandana, option 2. Okay. So quickly, everyone should answer this question. Whichever Vir protein you are confident about, just look at that and see which is the function. Is it 1, 2, 3, or 4? Okay, so let's see what is the answer of this question. Let's say I'm confident about Virg protein. I know Virg is the protein which is involved in activation, or I should say induction of Virg genes. So simply, if I know that A function, that is Virg function, is induction of Virg genes, I know the correct combination is A4. And now if I look at the options, only in option 2, A4 is given. Right? So simply, even if you know about the single one Vir protein, you can answer this question. But for safer side, keep it 2. Let's keep it 2. So like similarly, you can go for Vir D2, Vir B1, Vir F, right? So here I have the list for all the functions of the Vir proteins. Make sure that you revise because we can expect a question on the functions of Vir protein. So even the statement based question can be asked, or the question match the following type question can be asked. Okay, so you need to be ready for both the type of questions. But for that, to answer such questions, to attempt such questions, you need to be thorough with the function of the Vir proteins. Okay, so like Vir-F, it is involved in protein degradation. So if we see the option, like if you see the option third, it's about destruction of proteins containing tDNA complex. So yes, we can clearly say that the D and the third, it's the correct combination, right? So that is why the answer is option 2. So very good, you all have answered it correctly. Moving on to next question. So this is question number 3. Please go through it. Now you can see it's a graph-based question. Agrobacterium-mediated transformation was used to generate transgenic plants using a construct with a selection marker gene X and a passenger gene Y. Expression levels of Y protein in 8 independent transgenic plants are given below. So these are the 8 transgenic plants, and their expression level of the gene Y is given on y-axis. So following could represent probable reasons for the observed variability in transgene expression level. A, position effects on passenger gene. B, transgene silencing of the marker gene. C, variation in copy number of messenger gene, and D, messenger RNA instability of marker gene. Which one of the following combinations of above statements is correct? So to answer this question, you need to be familiar with these terms. What is position effect? What is copy number? What is gene silencing? Right? So analyze the graph carefully and tell me what is this graph represents. According to this graph, which of the following combination of statements is correct? Is it A and C? Is it C and D? Is it B and D? And is it A and B? Fourth option. Amala, very quick. Okay, option one. Nandini, Vishnu, Kriti, Ankita, Devanshi, Vishal. Okay, you all have answered option one. Polaris, Sachi, Aditi. Option one. Okay. Vanna, you are saying option four. Fine. Kaveri, Sucheta, Vijay, Sonali, Adesha, Garima. Okay. Option one. What we can infer from this graph? Selva and Jaya, option one. Okay. See, one thing is very much clear that the expression level is not uniform, right? The transgenic plant, like the expression level of Y protein in fifth and eighth transgenic plant is more in comparison to three and others, right? So, what could be the reason for such high expression level or such variation in expression level in all transgenic plants? So, what is position effect? If you need to choose the correct combination, you need to understand these terms first, right? Like, what is a position effect? So, basically, when we talk about position effect, it means the difference in expression level due to difference in place of integration of foreign DNA. Even in genetics, also, you study about position effect. Like, for example, in translocation, when a gene from one chromosome, it moves on to another chromosome, right? The gene from A chromosome moves on to the B chromosome, right? So definitely it comes, it interacts with the other gene. Now the enhancers which are regulating the neighboring genes will definitely affect the expression of the gene which has been translocated, right? So here also the position effect, where the position of the gene, the integration of the foreign DNA took place at different take place at different places. So definitely that will affect the gene expression. And when I say it affects the gene expression, so option A, like you can omit two options out of four. So make sure when you're solving such questions in exam, try to omit two options out of four. So that means A of statement is given only in option 1 and 4, not in 3 and option 2. So these two cannot be the answer, right? So to save the time in exam, make sure that you omit two options out of 4. This should be your strategy while solving the question. Okay, I know you all can solve, but the point is in how much time you solve that matters, right? Now let's check other statements. So one is position effect. Then one more term is there, which is copy number. So copy number, that is instead of one copy, more than one copy is being inserted, right? It can be integrated. Now definitely more the number of copies, more the copies of a gene are there, definitely it will affect the level of expression. So position effect and copy numbers are playing a very important role, and this is the reason statement A and C are correct. Now, if you see other two options, which is about the transgene silencing and messenger RNA instability, So they are about marker gene and not the passenger gene. So first of all, you need to pay attention that whether this silencing is been about the marker gene or passenger gene. So nothing has been mentioned about the passenger gene here. It is about the marker gene only. And in the question, the expression of Y protein is asked, right? But even if the question is asked in different way, okay, maybe the transgene silencing or messenger RNA stability of passenger gene would have been asked, right? So you need to be prepared for that question also. So you should know that if messenger RNA instability is taking place or gene silencing is taking place, then definitely we will get the uniform, like the uniform expression will be there. Because definitely messenger RNA instability or gene silencing will decrease the expression, right? And you will be able to see this uniform result or expression in all the transformants. But as in this question, it's not about marker gene, it's about passenger gene. So correct answer is option 1. A and C statements are correct. So most of you have answered it correctly. Very good. Almost everyone has answered correctly. Now moving on to next question. Okay, just a minute. Let me. Okay. This is the next question. Please go through it and tell me what is the correct answer. Vandana, I hope is it clear why the answer is option 1 for the previous question and not 4? So I'm really glad that you answered, because this is how we learn, right? We make mistakes and we learn. But the important thing is we should correct ourselves, right? Okay, so question 4 is about transgenic approaches. Some of the following transgenic approaches could be used for functional characterization of endogenous genes in plants. A. Transformation using a binary vector containing a strong enhancer element and lacking the right border of tDNA. Transformation using a binary vector containing promoter-less reporter gene sequence and selection marker gene cassette within the tDNA. Statement C. Transformation using a binary vector containing only a strong enhancer element and a selection marker gene cassette within the tDNA. And D. Transformation using a binary vector lacking reporter gene as well as both left and right borders of tDNA. Okay, K18, you can ask your doubt here. Others, you can solve this question. Which one of the following combinations can be used? Now, to answer this question, you should know what is a binary vector, and also you should know about the importance of left and right borders, and also about the reporter gene or enhancer element. You need to be familiar with these terms. Okay? What is a binary vector? Who can tell me? What is a binary vector? Let's revise this concept. What is a binary vector? Okay, B and C or option 2. Fine. So I can see the answers. Okay, so Cheta option 1. Fine. Vishal, co-integrate is different. Binary vector is different. Yes, you are right, K18, that binary vector, it's a standard tool. You are right. Correct, Nandini. They are kind of shuttle vectors. Okay, so I can see some of you have answered, majority have answered option 2, but some of you have answered option 1 and 4 also. Okay. Thank you, K18 ABI, for the response. Yes, I can see your answer. That's a standard tool in transformation of higher plants mediated by agrobacterium tumefaciens. Yes, you are right. But to be more precise, binary vector is what? It's a pair of plasmids, helper plasmid and disarmed tumor-inducing plasmid. So I hope everyone has answered the question. So I can now move on to the explanation and the answer. Okay, some of you are still answering. Fine. So I'll wait for a few minutes, few seconds, in fact. Anshu, okay, option 2. Fine. Let's see if your answer is right or not. Sachi, right border is most essential. Okay. See, these are very small facts, small points that you have to focus, right? You still have time. Make sure that you revise these important facts from this particular topic. So let's move on to the explanation. So those who have answered option 2, they are right. The correct answer is option 2 only. So I was telling you about binary vector. So see, binary vector is a pair of plasmids. So one is a helper plasmid, okay, the one which carries the weird genes, and another is disarmed TI plasmid, okay. So we call it as disarmed because it does not contains the oncogenes, okay, it does not contain the oncogenes. And the most common or the first binary TI vector which was developed was PBIN19. So you can see it has left repeat and right repeat along with tDNA. And then the marker gene is also there, that is kanamycin. Now, we can test endogenous gene of a plant. We can use two approaches. So either you can transfer and integrate a promoter-less reporter near the gene in question, right? So that you can see the level expression of the endogenous gene, or you can even integrate enhancer element with specific selection marker. Both the approaches can be used. But the catch here is that you need right border. Transfer of the components, these components into a plant cell using TI plasmid based approach, you need right border. Right border sequence is necessary for transfer and integration to plant genome. Please note this point. Okay. And when I say right border is important, so if you see at the A statement, it says lacking the right border. So if the right border of tDNA is missing, this is not the correct statement. Similarly, in D also. First of all, it is mentioned lacking reporter gene and left and right border. So again, this is incorrect statement. Because you need to, if you want to study the gene function, you need a reporter. Fine? And this is the reason that the answer is option 2. So this is all about unit 12. I know there are many concepts, many questions are there, but because of time's constraint, I can discuss only few, right? So I'm sure now you know how you need to revise for this unit 12, right? You know, specifically you need to emphasize on Ver proteins, you need to emphasize on transgene, right? Agrobacterium mediated gene transfer process. So now moving on to unit 13, that is methods in biology. Now, tell me, what is the first thing that comes to your mind when you go through the questions of Unit 13? Do you find them easy? I'm specifically asking about Part C. Okay, Part C questions. So, what do you think? Yes? Do you find it interesting? Do you find it hard? Do you find you are not able to solve? Tell me. Be frank, because then only I will be able to help you out. Yes? All of you find Unit 13 questions easy? Unit 13 is all about techniques, right? You study about radio labeling techniques, recombinant DNA technology, you study about immunotechniques, right? Even statistical methods, microscopy techniques, electrophysiological methods. So, although in part B, the questions are very direct. If you simply remember the principle and application of the technique, you can solve. But in part C, the questions asked are experimental, right? So I'm glad to know that you find it easy, you find it interesting. Okay, but some of you find it tough also. I'll absolutely agree with you, Sachi, because unless and until we are not familiar with the experiments, we are not familiar with the application of the techniques and even data analysis, specifically in case of flow cytometry, right? So it becomes very difficult to solve such questions. Okay, Garima, you find it tough. Fine. So thank you all for answering this question. So most of you find it interesting, but yes, tough and complicated sometimes. Okay, Ankita, Shivashi, quite analytical. Yes, right, Shivashi, and time consuming. Yes, you are right. So that means if you're specifically solving the questions of Unit 13, you have to make sure that in how much time you're able to solve a particular question, and you need to work on it. Okay, Adyasha, some are tricky, some are interesting, and need to have knowledge of techniques. You're absolutely right. So let's discuss few questions from Unit 13. And as we discuss, I'll tell you about the strategies of how to solve a particular question. Okay, so here comes the question number 5th. The diagram below represents a 2 kb insert successfully introduced between two BAMH1 sites of a 3.8 kb vector in the desired orientation. The Hind3 site on the insert and EcoR1 site on the vector is also indicated. If the insert was introduced in the opposite direction, which one of the following statements is incorrect? So, this question from cloning, go through the figure carefully, look what information is given in the question, and accordingly answer this question. Read the question carefully. If you are not able to understand it once, read it twice or thrice. And when you practice, you can underline the important points or you can write down separately the important points or the hints that is given in the question. Okay, Manoj, option 2. Let's see what others have to say. Option 1, Amala, okay. So it's a vector of 3.8 kb size and 2 kb insert is been introduced, which has two BAMH1 sites. It's already shown here. And even Hind3 site is present. But EcoR1 site is present on the vector. Okay, now I'm getting mixed answers. Ankita 4, okay. Nandini 3, Kaveri 3, fine. Devanshu 1, Sonali 3. So I really appreciate you all for answering the questions. Very good. Practice makes perfect, right? You all have to become perfect. Anshu, option 2, okay. Sucheta, option 2. Now, I'll use two words, and you think that based on these two words, you have answered the question or not. The two words are opposite direction, and second word is incorrect. Opposite and incorrect. Think about it. In the question, it is mentioned if the insert was introduced in opposite orientation. What do you mean by opposite orientation? If I have a sequence A to B, opposite orientation means B to A. Right? So here, if the correct orientation is 0.8 and 1.2, the opposite orientation will be 1.2 kb will be here and 0.8 kb will be here. Now, definitely to answer this question, you have to go through all the statements. But you don't have to choose the correct statement here. You have to choose the incorrect statement. So have you all selected the incorrect statement? Yes, Ankita, now you're changing your answer. Right? But in the exam, nobody will going to tell you this catch which is present in this question. Right? So make sure that you all read the question carefully. Okay? And when you read, try to find out the key points that you have to keep in mind. Opposite direction and incorrect statement you have to choose, right? So the strategy to answer this question is, please redraw the diagram. Okay, redraw the diagram based on the information given in the question. You're getting my point? So this is the correct orientation given in the question. You have to draw opposite orientation and you have to check the statements based on this opposite orientation, not on the basis of correct orientation. Clear? So what is the first statement? That if you perform the digestion with eco R1, it will linearize the plasmid into 5.8 kb. Is it correct? Let's check. So this is eco R1 site. If I just perform the digestion from here, just assume it's a thread, okay, a circular thread, and you cut it from a scissor. Cut it with a scissor at this point. So it will linearize. Now what will be the size? The vector was 3.8 kb, 2 kb was your insert. So total 5.8 kb. So that means option 1 is correct. And we have to find out incorrect, right? So that means this cannot be the answer, right? Option 2, which states the statement is that when you digest with BAMH1, it produces two fragments of 3.8 kb and 2.0 kb. So yes, it's right. BAMH1 site is here, another BAMH1 site is here. So if I cut it, so from here to here, it's 3.8 kb and this entire is 2 kb. So this is also right. Fine. But now, if we see option 3 and 4, if we perform the digestion, double digestion with eco R1 and HIND3, what we'll get? We'll get fragment of 2.1 kb and another of 3.7 kb. 1.7 and 4.1 you will get, but that in the correct orientation. But in the question, you don't have to answer. You don't have to find the answer according to correct orientation. You have to find the answer according to opposite orientation, right? And therefore, answer is option 3. Okay, so my point is, go through the question carefully. Okay, these words, correct, incorrect, true, not true, mentioned in the question are very important. Okay, pay attention to them. Fine, moving on to next question. But before that, I would like to tell you about our upcoming batch. See, I'm really glad to see all your answers coming. Most of you are answering correctly. But if you need any help, Biotecnika is always with you. And if you feel like you need to join the upcoming batch for preparing for the next exam, you can join the Shaurya batch. Okay, it's starting from 4th March. And these are the contact details. Okay, you can contact us using these numbers. So, are you all ready? I'm sure you are not tired. Ready to see the next question? Yes? Are you all excited? Yes or no? So, moving on to next question. Okay, Ankita, not tired at all. Glad to know. Keep it up. Ready, Amala? That's great. Polaris, okay. Thank you. Thank you, everyone. So let's solve the next question. Following are the typical dot plots of cytometric data obtained from peripheral white blood cells. R1, this R1 in plot 1 shows the selection window of cells which are further analyzed based on immunostaining. Plot 2. Analyze the given data. Choose the correct option representing the type of cell population in plot 1 selected and segregated in various quadrants of plot 2. Flow cytometry. This question is from flow cytometry. Very important topic from subunit 13B, immunotechniques. Definitely a question on dot plot analysis is asked in exam. So you can only answer this question if you know to analyze the dot plot data. Right? This was the question asked in year 2020. And even in unit 13, like unit 13 is complicated because sometimes the topics from unit 13 are merged. Like a question is framed by merging the topic from unit 13 and with the topic from unit 3 or unit 4, like flow cytometry with cell division from unit 2, right? Or immunology from unit 4. So that makes unit 13 a little bit more complicated. But with proper guidance and with proper practice, you all can solve questions. And you are actually doing it. So I'm really glad to see the answers coming. So majority says option 1, even few have to, some of you are saying option 4 also. Okay. So I'll tell you a very easy trick over here. Okay. First, I'll tell you what flow cytometry is all about. And then I'll tell you one trick that you have to, or one fact that you have to keep in mind. Okay. That how to analyze the dot plot. So basically, what is flow cytometry technique? Flow cytometry is a powerful tool that help you analyze multiple parameters on the cell. What are these multiple parameters? First of all, you can identify a particular cell based on the antigen or the marker present on a cell surface, right? Like now, the scientists, the immunologists, they know that if a particular cancer is present, then like a blood cancer or lung cancer, so they can identify a particular cancer based on the type of cells present, and we can identify a particular cell by detecting a specific antigen on their surface. Now, using flow cytometry, you can analyze multiple parameters. Immunologists, they analyze multiple parameters like size, complexity, or I should say granularity, right? And even there is an advancement in flow cytometry technique, which is called as fluorescence activated cell sorting technique. So using FSCS, we can even sort the cells like B cells from T cells, right, from a mixture of cell population. So here in this technique, basically the principle of this technique is immunofluorescence, where based on which cell we have to detect, so we'll target the antigen, and to this antigen, for this antigen, we'll use a specific antibody, which is labeled with a fluorochrome. So immunofluorescence is the basic principle of this technique. So as you can see here in this figure, that mixture of cell population is being injected into the flow chamber. Now these cells, they already, before injecting it into the flow chamber, already we have performed this antigen antibody interaction. Like whichever cell we have to detect, based on what antigen is present on that cell, we have used a specific antibody labeled with a suitable fluorochrome dye, fluorescent dye. It can be FITC, it can be phycoerythrin, it can be Texas red. Okay. Now the characteristic of this flow chamber is that here the cells are aligned in a single file and they just pass one cell at a time. And as they pass, we use a laser source which pass the light, and as the antibodies are labeled with a fluorochrome, these fluorochrome absorb the light and emit light of longer wavelength, which will be detected by the detectors. So based on what fluorochrome we have used, according to its excitation and emission spectra, we use the laser source as well as detectors. So we can identify a particular cell. And even if you want to detect, if you want to determine the size and complexity, for that you use two type of detectors. One is forward scatter, another is side scatter detector. So forward scatter detector is one that indicates cell size, and side scatter indicates complexity or granularity. So how it detects cell size? Like for example, one cell is smaller and another is larger, right? So if the light is passing like this, so if a cell is small, only forward scattered light will be this much. But if the cell size is bigger, the light scattered will be, forward scattered will be this much difference, right? So detectors determines this and help us in detecting the size. Similarly, if the granules are there, like white blood cells, we have granulocytes, right? So those cells which have granules, they will scatter the light sideways more, and side scatter detect these side scattered light, and more the granules, more will be the light scattered. So it indicates about the granularity, right? So here I have this figure I have taken from Cubase, and if you see this diagram, this figure carefully, you will understand that this question was directly picked up from the Cubase. From this data, the question was framed. From this dot plot, the question was framed, right? And this is the reason in our every class, we always motivate you to refer the standard books. You should never leave the habit of reading the standard reference books. Okay. I'm sure only few teachers are there who will tell you this, but in Biotecnika, we always encourage you to do that. So I'm sure now you realize the importance of going through the reference books, right? Because you all are future researchers. You will be going through, you will be studying a lot of research articles, review of literature. So you should never leave the habit of reading books. Okay. So let's see what this dot plot is all about. So here you can see this is the forward scatter and this is side scatter, right? On x-axis, you see the forward scatter data signal, and y-axis side scatter. So if you have studied about neutrophils, lymphocytes, you know that neutrophils are larger in size than lymphocytes. So obviously, as the forward scatter indicates size, you can see that the lymphocyte size is much smaller than neutrophils, right? Just imagine it to be a number scale. So obviously the values are increasing from left to right. And if we further analyze these lymphocytes in detail, right? So here it's the dot plot of the T cells, the T cells, one which is bearing CD8 and another which is bearing CD4. CD4 bearing T cells and CD8 bearing T cells. Now, I told you in the beginning, I'll be telling you one fact or one important trick here, that what you have to do is, like, to remember about or to understand the dot plot analysis, you have to simply draw a square and divide it into 4 quadrants. Always remember the first quadrant is double negative. Okay. So double negative means, like, here you are just first label it. So here we are measuring the fluorescence of T cells, CD8 bearing T cells, and here CD4 bearing T cells. So obviously this is double negative. That is, here the signal intensity will be, fluorescence intensity will be very low. And this is the quadrant for those cells which lack both CD4 and CD8. Okay. So always remember A quadrant, double negative. Now B quadrant, obviously, as the signal intensity for CD8 bearing cells is increasing from left to right, this is the origin point, left to right. So here you will represent it as CD4 minus CD8 plus cell. Okay, the signal is for CD8 plus, that is CD8 bearing T cell. C quadrant, similarly, it will be for CD4, but CD8 minus, that is those T cells which have CD4 but lack CD8. And D quadrant will be double positive, that is for those cells which will be having both CD4 and CD8 also. So this particular data was given in the question. So quickly you can say that A and D, A is double negative and quadrant D should be double positive. And simply on this fact, you can answer this question. And this combination is only given in plot 1. So answer is 1. So I'm sure now you will never forget this concept. Okay. Moving on to next question. A researcher is investigating structural changes in a protein by following tryptophan fluorescence and by circular dichroism. Fluorescence and circular dichroism spectra of a pure protein were obtained in the absence of any treatment. So A represents absence of any treatment. B, that is in the presence of 0.5 molar urea. C, that is adding acrylamide, a quinter of tryptophan. And D, upon heating. So data is shown below. So you have to choose the correct statement. Now this question is from subunit 13C, spectroscopic biophysical methods. Question on spectroscopy. So from this particular subunit, you can expect a question, numerical based question on Beer-Lambert's law, or statement or graph based question on UV visible spectroscopy. And even you can expect the questions on, like, instead of giving one spectroscopy, they can give you two spectroscopy, and they can give you some data, and they can ask you to find out the correct answer. So Mahima, yes, quadrants you need to remember with respect to the previous question. You are right. Labeling, quadrant labeling, you have to remember. Okay, so for this question, I'm getting mixed answers. Some are saying option 1, some are saying option 2. Fine. Go through this graph carefully. Sachi, option 1. Okay, Vishnu 2, Ankita 2, Vishal 1, Devanshi 1. Okay, Nandini 2. So I think there's a confusion between option 1 and 2 because nobody is saying 3 and 4. So I'm glad that at least out of 4, you are able to omit 2 options. Good. So option 1 is CD is more sensitive to structural changes than fluorescence. Option 2, fluorescence is more sensitive to structural changes than CD. Third option, both methods are equally responsive to structure changes. Or option 4, that acrylamide alters the secondary structure of the protein. So you have to remember the principle, the application of these techniques, spectroscopy techniques. Okay, tell me, circular dichroism is what kind of spectroscopy? Who will tell me? Is it absorption or is it emission? Basic concept. Let's revise. Circular dichroism is an emission spectroscopy or absorption? Vishnu, very good. Correct. See, these points should be on your tips. Okay? Fluorescence, when you see fluorescence, when light is emitted of longer wavelength, right? So fluorescence is emission, but circular dichroism is absorption. Please note, Those who have answered emission, please note. I'm sure now you will never forget this concept. I'm glad you answered. Very good. So circular dichroism, it's an absorption spectroscopy. Okay, it is based on differential absorption of left and right circularly polarized light. So it's an this particular absorption of left and right circularly polarized light is the characteristic of the optically active chiral molecules. And circular dichroism, we always, like, it is often reported in degree of ellipticity. So yes, I think the confusion is between option 1 and 2. So let me explain you the answer. Okay, let me explain you, and then you'll better understand why the answer, why a particular option is a answer. See, first of all, always keep in mind whenever such graph based questions are asked, don't think out of the box. Just focus on the graph, nothing else. Okay, here you don't have to use Beer-Lambert's law, you don't have to apply any other information. Simply compare the graph. What do you think? If I see the graph for fluorescence, right? Don't you think variations are there? Like for A, we are getting this fluorescence intensity. For C treatment, we are getting this intensity. For D and for B, fluctuations. There is no uniformity, right? But if we look for the data for circular dichroism, you can see that it's more or less like same, right? C, B, A treatment, it's same. Not much variation, right? So what you can infer, fluorescence is more sensitive or circular dichroism is more sensitive? One thing is very clear, both are not equally responsive. So 3 cannot be the answer. And here we have to look at the effect of the treatment. So acrylamide is a quencher, which decreases the fluorescence intensity of the tryptophan. But what do you think, which is the most appropriate option? Yes, it should be 2. Right? Although we have used different treatments, but remember, ellipticity graph is similar for all the treatment as compared to fluorescence, which have different graphs for different treatments. And this makes fluorescence more sensitive to CD. Okay, and therefore answer is option 2. So I'm sure now it's very much clear. There is no more confusion between option 1 and 2. So those who have answered option 1, is it clear why it will be option 2? Mahima, right. More fluctuation in fluorescence. You are right. Fine. Moving on to next question. Question 8. A protein undergoes post-translational modification. In an experiment to identify the nature of modification, the following experimental results were obtained. So A, protein moves more slowly in an SDS page. B, isoelectric focusing showed that there was no change in PI. And C, mass spectrometric analysis showed that modification was on serine. So modification that the protein undergoes is likely to be phosphorylation, glycosylation, ubiquitination, or ADP ribosylation. So now to answer this question, you should know what are different post-translational modification. You should know the principle of SDS page, isoelectric focusing, and also you should know about mass spectrometry technique. So what do you think is the correct answer? What is the modification that the protein undergoes? Now again, here you have to solve step by step. Rule out the possibilities. And you can only rule out when you know what happens when phosphorylation takes place, when glycosylation takes place, when ubiquitination takes place, and when ADP ribosylation takes place. Okay, Rajrupa option 1, Sachi 1, Vishal 1, Ritika option 4, Manoj, you are saying glycosylation. Okay. Jakka option 4. Fine. Devanshi 1, Amala 1. Okay. Aditi 2, Nandini 1. So I mixed answers I'm getting. Some are saying option 1, some are saying option 2, and even 4 also. See, always attempt this question. If such type of question is asked in exam, only answer if you are 100% confident. Okay, otherwise leave it. So majority saying option 1. What happens in phosphorylation? Phosphate group is attached, right? Just pay attention on the results. Protein moves more slowly. So you know SDS page where you can separate the proteins according to the molecular weight, right? So if it moves more slowly, that means it must have a high molecular weight. This is the one hint that we got. Second, no change in PI, right? So isoelectric focusing, PI, isoelectric point, when the net charge on protein is 0. And mass spectrometry technique, which measures the mass to charge ratio of a peptide. And it shows that modification was specifically on serine. So that means, first of all, you should recall here that if phosphorylation occur, glycosylation occur, ubiquitination occur, it occurs on which amino acid residue? And if it takes place, if modification takes place, does it cause any change in the, like, does it cause any change in the PI? So SDS page, you need to know the principle of this technique, isoelectric focusing, mass spectrometry, and most importantly, you should know about the post-translation modifications. So here I have added some post-translation modifications. Nearly every protein in a cell is chemically modified after its synthesis on a ribosome. So like acetylation can take place, right, on the N-terminus. Then phosphorylation, it specifically take place at serine, threonine, tyrosine, histidine residue. Then similarly, hydroxylation of proline and lysine residues in collagen takes place. Then methylhistidine, that is methylation of histidine residue in membrane receptors takes place. Then O-linked, N-linked glycosylation is there, right? So you need to be familiar with these post-translation modifications. Then when we talk about SDS page, so SDS being an anionic detergent, it denatures, it breaks the non-covalent interactions between the subunits and provide a uniform charge to the protein. So all the protein has a uniform negative charge. So basically, when you're performing the separation, when you're loading the protein sample, the separation is on the basis of the molecular weights, right? Those proteins which have high molecular weight, they will move more slowly. Like as you can see here, myosin, right? And even if you use a molecular ladder, using that you can determine the molecular weight of an unknown protein also, right? And what happens in isoelectric focusing? We provide a pH gradient and then you add the sample on the top, you perform the, you apply the electric field. So proteins are separated and they, when they reach to the isoelectric point, the net charge on become 0, right? So they stop moving. So as in the question, it is mentioned that there is no change in PI. But if you see phosphorylation, if phosphorylation occurs, we know that a negative charge is added, right? It will definitely change the PI of the intact protein, right? It shifts it to the acidic side. And therefore, phosphorylation cannot be the answer because according to the result of isoelectric focusing, there should be no change in PI, but phosphorylation causes changes in PI. Now, ubiquitination. So definitely if a ubiquitin is added, there is an increase in 8,800 Dalton. But this particular modification takes place to lysine residue. But mass spectrometry techniques tells us that modification was on serine. So again, this cannot be correct. Then ADP ribosylation. It is initiated by transferring one ADP ribose moiety from NAD plus to side chain of aspartate, glutamate, arginine, lysine, serine, and cysteine residue. So obviously, the phosphate moiety in ADP ribose will bring a huge amount of negative charges to local environment. So again, this cannot be the answer. And yes, glycosylation. So intact glycans, high molecular weight modification will take place. And as I told you, Olin glycosylation occurs on serine or threonine. So SDS page tells us that the protein moves slowly. That means it has a high molecular weight. And the modification was on serine. So this also confirms the answer. And therefore, answer is option 2. Clear to everyone? So you have to go through all the techniques. The basic principle you should know, you should learn, and you should try to connect all the points, all the information, post translation modification related to these techniques. Okay, quickly moving on to the question 9. The most important property of any microscope is its resolution. So this is from microscopic techniques. And the resolution can be calculated from the formula. This is the Ernst-Abbe equation that is given in the question. D equals to 0.61 lambda upon n sine alpha, where D is the minimum distance between two distinguishable objects, lambda is the wavelength of the incident light, alpha is the angular aperture, and n is the refractive index of the medium. So given below are a few suggestions to improve resolutions of a microscope. So what do you think? If you decrease the wavelength or increase the wavelength, what will be the effect on resolution? Similarly, if you use oil of higher refractive index or lower refractive index, what do you think should increase the resolution? Okay, some are saying option 4, some are saying option 1. Okay, majority says option 1, fine. Now here you have to go through all the statements. Now see, A and B are contradictory and C and D are contradictory, right? So what do you think can be the answer? Now to answer this question, you need to be familiar with the terms. What is resolution? What is refractive index? What is numerical aperture? This n sine alpha is called as numerical aperture. Okay, A and C, that is option 1, Vishal, fine. What about others? I'm sure you all must have seen the microscope in your colleges or universities, and even some of you have used it. Yes, Sachi, you are right. So recall all the concepts here. So majority is saying option 1. Let's check. So see, first of all, you need to know what is resolution. Okay. Resolution is the ability of a lens to separate or distinguish between small objects that are close together. Right. So here, if we have like here, the sample is placed. So obviously the light which is reaching onto the objective lens, you can see it is forming a cone of light. Okay. It is forming a cone of light. I'm sure you all have seen ice cream cone, right? So it's forming a cone of light. Now always remember the angle of cone of light that can enter a lens depend upon the refractive index of the medium in which the lens work, as well as upon the objective itself. Okay. So theoretically, the refractive index for air is 1. Okay. So I told you numerical aperture, that is numerical aperture, this expression, this n sine alpha is a numerical aperture, right? So it depends upon the angle alpha and refractive index, right? So alpha is the maximum half angle of cone of light. That is alpha. In some authors prefers to use theta or mu in place of alpha. So don't get confused if in the question theta is given or mu is given. Now, theoretically, the refractive index of air is 1, and the maximum possible angle of uh, is 90 degree. So you all know that sine 90 degree is 1, right? So that means numerical aperture is 1 here, right? But if we want to increase the numerical aperture, we have to increase the refractive index. Now you can see that as the light is passing from the sample to the lens, in between you have some air. Now the light is passing from glass to air, to the lens, to the air, and then to again to the lens, right? But in between, because there is a change in medium, from glass to air, some of the light is refracted, bend. Refractive index is light bending ability of a medium. So here medium is air, and because of this, the light is lost. So why not to change this medium with something which have the similar refractive index as that of glass? So glass has a refractive index of 1.5, and even oil has a refractive index of 1.5, right? So if I again put the value, now the refractive index of oil is 1.5, and sine alpha, that is alpha is 90 degree. So again 1, so 1.5. Sine 90 degree is 1. So numerical aperture value is 1.5, right? So the only practical way to raise the numerical aperture and to achieve higher resolution is to increase the refractive index with oil, which has the same refractive index as glass. So remember, higher the resolution, that is lower d value, can only be achieved with n having larger value, refractive index having larger value, and lambda having smaller value. So lambda, that is the wavelength of the light, and we always prefer the blue end of visible spectrum, that is around 450 to 500 nm. So answer is option A, and the correct statement is A and C. And now moving on to the last question. So let's discuss one numerical based question also. So usually from radio labeling techniques, the questions are asked on different forms of radiation. But if you see this question, it is about counts per minute. So 1 curie is defined as quantity of radioactive substance having a decay rate of 2.22 into 10 raised to power 12 disintegrations per minute. How much of counts per minute CPM will be recorded for 100 microcurie of 32P when measured in scintillation counter working at 50% efficiency? So quickly answer this question, students. I really apologize. I have extended the this session, but I think it's really important that you should revise all these concepts. And I try to bring the variety in the questions. I try to put on the questions from different subunits of unit 13, right? But because of time constraint, I can add only few. So please go through this question quickly and let's solve this question. So you need to understand, you need to recall what is the relation, how you calculate the efficiency, what is the relation between CPM and DPM. So again, here I'm getting mixed answers. Option 1, option 3, option 2. Okay, majority says option 2. How? Check. What if the option 2 is not correct? See, the difference in option 1 and option 2 is only in this value, 10 raised to power 8 and 10 raised to power 6. So make sure that you all are answering, you are solving correctly. Similarly, in option 3 and 4, it's the difference in 10 raised to power 6 and 10 raised to power 8. Okay, option 1 and option 2. So scintillation counter is an instrument that we use to measure the radioactivity. So what is the efficiency of the scintillation counter is 50%. So how much of counts per minute will be recorded? Okay, it's a good choice, Ankita. If you are not 100% sure, you can skip this question in the exam. Yes, you can do that. The right approach is this only. We should not fail in this practice of solving all the questions, especially if we are not 100% sure, right? Okay, so I think all of you have answered. Let's check the answer. So basically, the answer is option 1. See, the given in the question, it's given 1 Curie is 2.22 into 10 raised to power 12 dpm, and dpm is given 100 microcurie, and efficiency is 50%, which I can write 0.5. Now, efficiency is what? It's the ratio between counts per minute detected by the instrument and actual disintegrations per minute. Okay. So I can write efficiency is equals to CPM by DPM. And as we have to calculate CPM, I can simply rearrange, right? So CPM is equals to DPM into efficiency. So DPM is 100 microcurie. So I can write 100 into 10 raised to power minus 6. So you need to work on conversions also, conversion of units, right? Then 2.22 into 10 raised to power 12 into 0.5, that is efficiency. So always try to solve these raised to power terms separately, okay? So 10 raised to power minus 6 and 10 raised to power 12. So you will get 10 raised to power 6. But I can add these two 0, so 10 raised to power 8. So what will be the answer? When you multiply with 0.5, it will be 1.11 into 10 raised to power 8. Okay. So with this, I have come to the end of this session. So exam is like most of you, like the next 2 days will be very much crucial, right? And I wish you all the very best. I hope this rapid revision round series would have been very much helpful for you. It made you revise the key concepts related to all the units of CSIR net exam. And after the exam, we'll be coming up with more sessions. Remember, you are not alone in this journey of CSIR net exam preparation and even after that. Biotecnika is always there with you. Okay. So you can, for the updates, you can subscribe to our YouTube channel. You can join us on our Telegram group, and even you can download Biotecnika app. Okay. So all the very best for your exam. Thank you for now and take care.